Okay, 안녕하세요. 나는 시강wei. Hello everyone. Hello Korean friends. This is Shi Gangwei from GenScript ProBio. I'm the director of the process development and manufacturing for plasmid and viral vector in gene and cell therapy. I have more than 10 years in developing and manufacturing of biologics and cell therapy products. Today, I bring the topic about how to improve plasmid and the lentiviral vector manufacturing in gene and cell therapy. So gene and cell therapy is a type of personalized medicine that can treat a number of diseases which traditional treatment methods are not effective or cannot be applied. Plasmid DNA and the lentiviral vector is widely used as a delivery vehicle in GCT products, but process development and manufacturing of plasmid and viral vector are facing many challenges, such as high cost, lack of clinical grade supplier, scaling up, and low yield. Plasmid DNA can be used as a raw material to produce different kinds of viral vectors, such as lentivirus, AAV, and other types of viral vectors. High production yield in the range of milligram to gram level. Gram level is a must for by pharmaceutical industry. For the manufacturing of lentivirus, the capacity to produce clinical grade lentivirus in large scale is the most significant limitation for the industrial development. GenScript ProBio lays out Plasmid Platform and viral vector platform for gene therapy and cell therapy. GenScript ProBio has a strong experience in manufacturing plasmid as a different purpose, such as a raw material, as a drug substance, or DNA vaccine as a final product. For the viral vector platform, GenScript ProBio has a mature adherent system to produce lentiviral vector. Besides, viral vector manufacturing with a suspension system is also viable, which is not only solved the industrial bottleneck in large-scale manufacturing, but also reduce the COG. For the preclinical through clinical and even to the commercial supply, GenScript ProBio is matching forward to support more gene and cell therapy to success. So today in the global over than 650 products are in development or in the market. 75% of products in the clinical phase 1 or phase 2. So most of the projects are in the clinical stage. 12 of them were approved to the market. Most of the projects are using adenovirus, lentivirus, and AAV. All the three viral vectors are most popularly used in the gene and cell therapy products. A number of GCT products have received fast track designation, or fan track designation, or the breakthrough designation. So today I talk most about how to improve the plasmid vector and or the viral vector to how to process the process development and manufacturing process. Recombinant viral vectors are excellent tool for introduce of genetic material into host cells, like into the cell therapy like T cell or the NK cell or other cell therapy. There are many choices, and each viral vector has its place. Once you understand what you need from your viral expression study, you will want to understand how each viral vector compared to your goals. So different types of viral vector have different properties, have a different production system, like using the transfection system or stable cell system, or you also use the infection system to produce the different kinds of viral vectors. Also, different kinds of viral vector have a different capacity to deliver different size of the gene, and also different viral vector have different virus particle size from the 100 nanometer, and also different kinds of viral vector has a different gene integration, also the different immune response. So based on your project, you need to select the appropriate viral vector to deliver the gene of interest. So this is the classical viral vector manufacturing process. So currently in the worldwide, there are two classical viral vector manufacturing process. One is using the adherent system, another using serum-free suspension system. Each system has its own advantage and disadvantage. Adherent system is a mature viral vector production system to produce different kinds of viral vector, and but it also has a limitation for the low yield and the scalability. It is hard to scale up using adherent system. For the serum-free suspension system, it has the advantage of the high titer and easy to scale up, but it lack of the process stability. To date, there are many challenges in viral vector process development and manufacturing. For the so currently in different countries and regions, gene and cell therapy is a very hot study in the global wise. So currently in different countries and regions, there are unclear regulation expectation. Different countries have different regulation for gene and cell therapy products, but it currently rapid growth market. Currently, it is hard, is very difficult to obtain the clinical material for the clinical trial. Also, the viral vector is with low process yield, so we need to scale up to large scale to obtain the enough material for gene and cell therapy clinical trial. Also, the viral vector has a production has a highly variable analytical assays and also unqualified operation unit. It is hard to control the operation for the process. Also, the high cost of goods and lack of the clinical grade supply, large scale manufacturing for the early stage development or the later stage development clinical trial, and process development can early detect the large change during the process development. Also, from the for the viral vector process development, there are still a lot of checkpoints in the process development based on the viral vector its own property. For the very sensitive shear for the viral vector, also the temperature is very sensitive because for the viral vector is not suitable to store a long time for the viral vector, and also freezing thaw its sensitivity is very tricky because when you are using the viral vector, you need to avoid freezing thaw the viral vector. For the viral vector is too sensitive to salt. During the downstream purification, you need to consider the salt concentration to the viral vector function titer because high salt concentration may impact the function titer of the viral vector. Also, the lung infection particle for the final product. Also, because currently most of the lentiviral vector or the AAV are using the transient production potential for the batch-to-batch variation and scalability. It is very hard. It is very challenging to scale up from small scale to large scale for the viral vector manufacturing. For the GMP the manufacturing for the lentiviral vector, you still need to consider a lot of points like viral vector design. You need to consider the IP, the promoter of the vector, also the gene of interest. Also, you need to consider because move to the later stage GMP manufacturing or BLA to commercial in future. You also need to consider the host cell line IP. The license for using the host cell line, the safety of the cell line, and also you need to get the high titer using the host cell line. For the manufacturing operation unit, the unit operation selection, the scale up, the scale you want to scale, you want the environment GMP environment control, limitation or removal impurities from the process. Also lack of the standardization, international standards, QC, also for the in-process control, product and batch-to-batch release, also the process validation for the manufacturing in future. GenScript ProBio is has a plasmid and viral vector process development and GMP manufacturing platform. GenScript ProBio has two platforms. One is plasmid, another is viral vector. currently, the platform can have the different types of the plasmid and the viral vector purpose. Professional provide different types of the plasmid and viral vector for the IND, IND filing and the GMP manufacturing for the clinical trial. So currently, in this platform, we have more than 30 plasmid and viral CMC and the clinical GMP manufacturing projects. 9 of them projects, we have produced the 9 projects for the clinical trial. And also 5 of them, we have get the IND clearance, get the IND approval from US FDA and China MPA. And we have 1 submission in the recent, and we'll get the IND approval in next month. About more than 10 ongoing CMC projects will file US FDA and China MPA in future. So gene therapy and cell therapy study is very hot in recent years. So, but it is currently still startup stage. So currently we have a lot of strategic collaborators to combine together to get the project successful, to get the project success. And we have a different the types of the service to cover the whole product life cycle, like a pre-IND CMC study and to file IND and clinical and commercial manufacturing, and also for the plasmid, different kinds of the different kinds of viral vectors. So in this platform, currently we have major in 4 platforms: cell therapy, gene therapy, mRNA vaccine, and DNA vaccine. So just take cell therapy as an example. We provide a plasmid system, different 4 plasmid system, process development and GMP manufacturing. Also provide GMP level lentivirus for the clinical trial. And for the gene therapy, we have the platform for the different serotype of AAV development and manufacturing, and both for the plasmid and viral vector manufacturing. Also for the mRNA vaccine, currently in the recent year, in this 2020, this year, we have developed a lot of mRNA vaccine against the COVID-19, and also DNA vaccine development for the final DS and DP development and manufacturing. For the plasmid platform, plasmid process development and GMP manufacturing platform, we provide milligram to gram level plasmid service. The fermenter of the plasmid production is ranged from 5 liter to 100 liter using stainless steel bioreactor and single-use reactor. In this platform, we are under fully regulatory compliance, and we have the high product quality control, and also for the this platform is with high yield, high recovery rate, and can reduce the cost for the manufacturing. Also, just as I said, we have a lot of reputation and track record in both in US FDA and FDA, and we have the confidence to bring the project to success. And this is a robust plasmid manufacturing process, including the upstream, downstream process. For the upstream process, we are using the high density fermentation process with a feed batch strategy. And it is a process with antibiotics free and animal component free. The downstream part is using the multi-step chromatography steps, and with the final step, fill and finish step, to produce the different purpose of use of the plasmid. This is a case study for our plasmid manufacturing. The left picture is a fermentation process. Just as I said, we are using the high density fermentation process. This process is antibiotics free and animal component free, and the downstream part with we are using the multi-step chromatography steps to purify the plasmid. And this is a release spec for the plasmid we produced, and this spec was used for different projects, and also get the approval from US FDA and China MPA. And this is a standardized release spec for the Plasmid. For the viral vector platform, we have both adherent and suspension platform, and we have the flexible technology and the scale at choice. And even for the adherent, we can maximum the capacity is up to 75 liter per batch, and the suspension system is up to 200 liter using the single-use bioreactor. And we also have the fully GMP facility to control the quality system. Also, we have the different strategy to control the cost for our customer projects. Also, we have in GenScript, we have a huge capacity for the demand from our customer projects. So the time is very can save the time for our customer projects. And this is a capacity for the current platform. We have totally 4 production line for the viral vector manufacturing, and 3 of them are using adherent systems, and 1 of them is using a suspension system. The scale is from 50 liter to 200 liter, and currently we can provide the preclinical CMC study and IND filing, and even to the early stage clinical trial to produce the viral vector material for the clinical trial. And we also have the plan in 2022, we will scale up the process and to provide commercial use. When the project move to BOA, to commercial stage, we can produce the material for the product when launched to the market. And this is a typical adherent culture system for the lentivirus manufacturing from the cell banking, and using the cell factory system to doing the cell factory system to doing transfection. And for the downstream part, you apply the multi-step to purify and concentrate viral vectors. And for the using the adherent using the cell factory system, there are a lot of open operation. So GenScript has developed a closed system and can avoid cross-contamination, and also fully automated operation for the multi cell factory operation. And it is easy to manipulate. Also, it is easy to scale up. And using this system, we can operate the inoculation, transfection, medium change, sampling, and harvest using this one system. And we can also easy to scale up using the adherent using this cell factory system. And this is the lentiviral vector release spec for the lentivirus. We have a total about 22 release spec for the viral vector. And this spec release table is we have produced clinical material in US FDA and China MPA, and this spec was accepted by US FDA and China MPA. It is the standard release spec for our platform. And also we have the suspension, serum-free suspension system for lentivirus production. We have the own in-house cell line system to produce the suspension lentivirus. This is a chemical defined serum-free, total serum-free system, and with a high titer, it is a 10-fold increase than the system. And the right picture is a typical procedure for the suspension lentivirus manufacturing. Use a wave and single-use bioreactor for the upstream, and the downstream use a UFDF and chromatography steps to purify and concentrate the viral vectors. And this is a comparison between a suspension system and adherent system and at GenScript ProBio. And for suspension, a serum-free suspension system in GenScript, we have a higher viral vector titer, and it is a 10-fold increase than adherent system. And also have the low risk about biosafety, because using this system is without a serum. It is a totally serum-free system, and it can control the biosafety. And also using this system have the benefit for the cost reduction. And and also if you scale up and using this sun-free suspension system, and it can about even to 25% cost reduction for using this system. And also for the, just I said, from the different scale from the self flask system to 10-liter bioreactor, or even to the 50-liter bioreactor, the process is very stable, and the cost is highly reduced for using this system, and also for the titer, for the recovery is also very stable from the different scale, from small scale to large scale. And in GenScript, we have different types of the service. And for the Plasmid platform, currently we have the very fast order delivery time, turnaround time for the order, and our customer no need to wait time. So we also provide 3 types of the bioreactor for the different field of use, also different study purpose, from the pro grade, GMP pro grade, or the GMP grade. 3 types of the different types of plasmid on the different core system, and for the different stage of the drug for the cell therapy products. And we also have the off-the-shelf Lentiviral plasmid. We have the ready-to-use hair plasmid system, and it can be faster used for the project. And for the three types of the plasmid, the first is the plasmid grade, and this grade plasmid we can have a capacity from milligram to gram level, and with critical QA control throughout the whole process, and detailed records and documentation for ensure the traceability. Also, this process plasmid is animal-free and antibiotic-free to reduce harm to animal and human body. And this process is high fermentation with high yield. And also, we can provide the plasmid DNA as a drug, or the drug substance, or the drug product. And we have the total package of the CMC study to file IND. So from the plasmid construction, cell banking, process development, and the quality study, batch production, and stability testing. So it is a very, we have a lot of track record and experience to help our customer to file plasmid CMC to IND filing. Also, we have the GM Pro plasmid. It is popularly used for the clinical stage, and we have the animal-free, antibiotic-free to reduce the harm, and to benefit the by safety. Also, very clear plasmid as a raw material for cell therapy in the clinical trial. In this kind of GM Pro quality system, we apply the phase-appropriate compliance tool for the quality oversight to well balance the cost, efficiency, and the compliance. And we have the capacity, maximum capacity is 100 liter to produce large-scale plasmid. And we also have when the project moves to the later stage clinical and commercial stage, GMP plasmid is required for your project. So GenScript have the facility in US can provide fully GMP level GMP plasmid from small scale to large scale, and it is fully on the GMP compliance. For the viral vector Lentivirus platform, we provide research-grade Lentivirus from the different scale, and the turnaround time is very fast for the project under the research stage, and the release spec is standard release spec for your projects. And also, we can provide Lentiviral vector CMC study for IND filing. So for the cell banking, process development, and analytical development, pilot production also includes a stability study. And the turnaround time is about 8 to 10 months for the whole package for the CMC. And in GenScript, when the project moves to clinical stage, GenScript ProBio can provide GMP level lentivirus and it is under fully GMP compliance, and the different scale from the even to the 2025 self factory, and also even to 50-liter single-use bioreactor based on your project. So it is fully under the GMP compliance, and for the is properly used for the clinical trial. And for the in GenScript, we have the because in gene and cell therapy is a very new area for the drug development in the worldwide, and in different development stage, we will use we adopt the phase appropriate compliance pack system for your different study stage. So currently we have the pack system to for your different stage of study. And just ProBio, we adopted the methodology and the philosophy as a per PDA TR 56 to set up the phase appropriate compliance system to ensure all production QC and GXP relevant activities in compliance with our project's expectation and their purpose and corresponding regulatory requirements. And ProBio has developed specific guidance to ensure the production compliance, produce high quality products to clients and cost efficiency. Also, using this pack can save the time and also save the cost also the quality can be guaranteed to provide to faster deliver the projects. Also, we have the fully quality management system and based on the ICH, FDA, MPA, and WHO. And our quality system was established based on the applicable regulation and standards, including 6 major subsystems: QA, QC, protection, facility and equipment, material and personnel. Besides the system, we to ensure compliance to and provide high quality product. We also have good quality culture from the management team or to the operation team. We take the quality as the highest priority goal and have a good GMP mindset and fully understand that quality is designed and implemented by the production and QC and the GXP team. And we also established the GMP documentation system. We have 4 level set up 4 level documentations: policy, SMP, SOP, and SOR. And also, this document was well managed in this GMP system. And for the validation management, we have established excellent validation and qualification system, covers the corresponding models, facility, requirements, and the qualification, instrument qualification, and electrical methods validation and verification, and the computerized system validation, CSV system, process validation, and cleaning validation. Also, for the customer quality verification, we have the total CQV system. We have developed a ProBio specific CQV program to ensure all the quality requirements from the MSA, QA, and changes during the PM communication and identified and tracked to meet before, during, and after your project production. So we also, for our current facility, and we have in GenScript, we have the hold the ISO 9001 certification, quality certification, and we also for this site and for this system, we have accepted the global-wise customer audits and also the third-party audits. And currently, all of them are satisfied with the currently system, and also with are satisfied with our quality and compliance system. And we also moved this project to the first step. And for our quality system, so because plasmid and viral vector are widely used in gene and cell therapy, So and also project at different stage can have the different quality system. So in GenScript, we have this spec system to do the physical product compliance, to do the faster treatment for your project. And also we have the fully security system and also the validity system to ensure the quality of the product you produced in GenScript ProBio. And this is the final summary for my side. And we have this gene and cell therapy vector platform. We can produce different types of the viral vector, and to assist the earliest, the most IND clients in clearance in China. And we have a lot of track record in China and the US FDA. And we also will fast have the IND filing in South Korea. And we also have the GMP manufacturing for the clinical trial in US and China. And we have line projects in the clinical stage. And in this platform, we have the excellent platform, one-stop solution from the plasmid to the viral vector, from the drug substance to the drug product, from your early stage development to the clinical to IND to clinical stage, or even to commercial BOA to commercial in future. And no wait time for your projects in GenScript platform. Also, we have the high quality with the QC specifications. Also, most important, we use the physical proprietary compliance to balance the cost and efficiency. It is under the quality requirements of your project during your different stage. That's all my precision. I welcome you to if you have any question, we are very happy to answer your question. Thank you, everybody.